As the slide says, I'll be talking about the activation of the photoactive yellow protein. This work was published in 2004 in a series of papers before that, and it's primarily the work of Gerrit Groenhof, who was a PhD student in my group in Groningen. And this actually works. Now, what the photoactive yellow protein is, it's believed to be a cytoplasmic photoreceptor, and basically you have this nice little salt-loving bacteria, which is a photosynthetic bacteria, and it needs as much light as it can, but it can't stand ultraviolet light, and so it spends its day gradually floating up to the surface, and then when it detects too much light, swimming down to actually protect its genome. And so it shows a negative phototactic response to blue light. And effectively what this protein is a timing mechanism. so it goes a normal tumble, swim, which is a normal bacterial way of moving through space. And the time between the random tumbles and the length of swimming is going to be controlled by how often this particular protein gets activated. Now the structure of the protein is known. It's got 25 amino acids, 125 amino acids. It's got a p-chimeric acid chromophore, which is covalently attached to a particular cysteine residue. Now, the basic nature of this photocycle is as follows, and actually we want to suppress fluorescence as much as possible. So Paul is very presumptuous to say that I will talk about fluorescence. But essentially, there is a photoabsorption because, as I say, this wants to detect light and control. So we go from a resting state to a photoactivated state. Then there is an isomerization around a particular double bond, which causes a redshift in the spectrum, where we get to the photoactivated red state, that's shown on the bottom. After this, the protein unfolds as part of its signaling process. And then finally, and it's actually the unfolded state which is detected as a part of the mechanism to tell it when to stop swimming. And then finally, we have the protein refold, and in fact that this particular chromophore can't isomerize in the protein, it must actually unfold, re-isomerize in the protein refolds. So this actually takes milliseconds, and certainly not the nanosecond which is appearing on some people's slides for the rate of protein folding. So what we wanted to do was to understand, firstly, how the photon absorption induces the isomerization of the chromophore, and how the isomerization of the chromophore induces the structural changes in the protein that actually leads to the signaling. Because, and this is really comes back to part of the thing which is being discussed in this conference. I mean, some people think that I pump energy into my protein and therefore it's going to unfold. I mean, because that was one of the things as well. We thought, well, we're actually making a major transition inside the protein. We're isomerizing it. This is also going to disrupt it. But proteins dissipate energy like crazy. And in fact, the energy which was released in this is actually totally non-relevant or irrelevant for the protein. So what we're actually going to do, and I'm sorry because I swapped some of these ones, the animation is out of order. Effectively, what we're doing here is that we're going to have some reaction barrier, and speaking to this audience, I feel quite ashamed to be presenting some of these things sometimes. So instead of actually having a barrier between our reactant and our product, in this case our product will sit at a slightly higher energy than our reactant, because this is going to be a photoactivated process, we're actually going to put it up on some excited state surface. On this surface, the actually isomerized state could be the lower state. The system will evolve at some point. We'll drop back down to the ground state, and hopefully the system will continue across to product. And what we're actually wanting to do during these calculations is to actually do a dynamic surface hopping technique, and so we will actually be calculating as we excite the system onto the excited state, and we go through the region of these conical intersections that Seth was talking about, that we'll be trying to calculate the probability of jumping back to the ground state and doing the whole process dynamically. And this is going to be governed by both the dynamics of the nuclei and also by how close these two surfaces come. So this is, of course, a mixed quantum classical simulation where we actually have a small region of our system, because, of course, the activation, isomerization, is going to be quantum mechanical processes, which is embedded in our classical environment. So effectively, actually, we've got one protein molecule, so 125 amino acids. It's in about 5,000 water molecules, SPC, in a truncated octahedral box. 6 sodium ions. We're dealing with the QM subsystem, which was 22 atoms. So basically, this is the the chromophore, the residues which lie above and below it, and also the residue to which it's actually covalently bound. We're going to do CAS-CF, and as I say, do a diabatic surface hopping algorithm, and we're describing the protein in terms of the Gromov 96 force field. Now this is really, so I know what CAS-CF is, although I think most other people here will do. So basically we're holding the occupation of a range of the orbital fix, and there is a range over which we're allowing this thing to undergo a full CI calculation. And if I can, I might actually get you to dinner slightly earlier. So what you actually see on this particular animation, it starts off in the ground state. We're then going to flash it, so we put it up to the excited state. This is a successful transition. I'm not going to show you another successful transition. So then we're actually going to get isomerization around this particular bond. As it gets to 90 degrees, it jumps back to the ground state, and then in some cases it comes back up. But in this case, you see that there is a successful isomerization and a rotation around this other bond. So just to give you some confidence that these calculations are totally unrepresentative, this is actually a final state from the simulation after we've actually seen this isomerization going on. And this, for comparison, is a low temperature X-ray structure of a trapped state where they believe they've actually trapped the red state, isomerized state, by crystallography, and you can see that there is actually almost an exact one-to-one correspondence between the positions of what we get as our final state from our mixed QM-MM calculation before the system's allowed to proceed further, and actually the proposed trap state, which was actually found by Getzoff and also some other people as well. Now, what we actually have in terms of this chromophore is that we actually have the phenolic group is actually deprotonated in the resting state. It's hydrogen bonded to a glutamic acid and also to a tyrosine. It has a charged arginine, which also sits just above this group, which actually helps prevent rotation of the group and also stabilizes its belief in this particular charge state. We're going to see rotation around this particular torsion B, and the isomerization involves both the rotation around the double bond, but also a repositioning of this thioester bond and the breaking of this particular hydrogen bond, which is there. Now what this just shows some results of a typical trajectory, just so we get the order of what actually happens in this particular reaction. So what we're actually following is the change in the dihedral angle of this of the double bond, and that you can actually see once it rotates to around about 90 degrees, we actually get a surface hop back to the ground state. This one remains stable, and then at a later time we're beginning to see the changes in the other dihedral bonds which are associated with the chromophore. So certainly the rotation around the double bond accords perfect. Now this calculation was repeated on this one about 14 times, and we can, by looking at the final cis-trans probability, we can get an estimate of the quantum yield. I know the statistics are not great for this. We could also, by actually looking for when we actually jump out of the local basin and go to this 90-degree state where we get most of the surface hops, we can also have a look to see how this compares to the fluorescent lifetime, because basically that we won't get fluorescence once it's fully rotated. So, if we actually see, compared to experiment, we're expecting a quantum yield of about 0.35, and we get a quantum yield of 0.3, if you believe statistics over 14 molecules. The other thing that we actually find in the fluorescent spectrum is you get a series of oscillations in the fluorescent spectrum. And if we actually relate this to the oscillations of our dihedral, we actually manage to get similar sorts of wiggles between what we'd expect to be between the two surfaces, and we get values of around about 1.6 to 4.8 hertz, and experimentally they're roughly the same order of magnitude as well, again to give some confidence in terms of the way the calculation goes. And if we actually calculate the fluorescence lifetime based on where this actually jumps out of this particular well, again we get a fluorescence lifetime which is in the same order of magnitude. I mean, these are really rough numbers, I mean, because our statistics are really poor and I never actually trust the dynamics of these systems within about a factor of a half to 1 to 2 anyway. You should be very careful with molecular dynamics simulations. We're actually dealing with things like SVC water. It has a diffusion constant which is roughly double what it should be. And of course this feeds through to many other things. The fact that you've got a constrained model, the fact you've got no real hydrogens in there in terms of van der Waals, all affect the dynamics of processes in solvent. And I should have changed that. I could see that I was working on it, that I was going to get back to that. To answer one of Ross's questions of what he would like to get, I mean, of what does a protein really do in this case? And one of the things a protein does is to actually trap the chromophore to prevent internal rotation. And I'll actually show you an example of that in a second so you can see it more clearly. I mean, one way of actually getting this isomerization would, of course, be to write the whole tyrosine ring, but that's actually not possible in the solution. It appears that the protein actually acts because, so what you actually find, I should actually do this in a little bit different order. If you actually run this calculation in vacuum or in free in solution, you find that actually you don't get the isomerized form. In fact, that either you get fluorescence or the system eventually just collapses back into the original one that it was in the first place. In the protein appears to both stabilize the excited state, so to shift the, to lower the energy of the excited state relative to the ground state, so the excited state at the moment is actually in red with the ground state in blue, and actually to lower this barrier which allows it to actually twist to a 90-degree minimum, so it tends to fall over, and then it's actually a series of bond stretching and skeletal changes which actually bring it close to this conical intersection where it then collapses down to the ground state quite efficiently. So in terms of actually what this does inside the protein, I think it's a little bit easier if we see it on this one, where the protein is actually being removed. So again, it's been flashed. The one in green you can see is the original structure, and so you can see there's almost no change in the position of the benzene ring or phenol ring. It's actually when this gets to 90 degrees, if you see the jump down to the ground state again. And in fact, this whole rotation, I don't know why that keeps on the difference between a Mac and a window in terms of PowerPoint, and of course it won't do it again, which I do believe is Bill Gates trying to make it that you should never use these things in reality on a Mac. And the Mac people trying to make sure that you actually use their one instead of Bill Gates' one. So what you actually see when this rotation actually occurs, there is a slight shift of the whole unit, which actually is not that there is a twist, it just pulls away, and so it's slightly further away from these hydrogen bond donors. So basically I've just shortened that by a very small amount. Okay, so if we actually see what actually happens to the protein once it's undergoing this twist and isomerization, and what you see is nothing. Okay? This whole isomerization process has no effect on the protein at all. And so you can actually see that we can propagate both systems, and we can propagate them actually for a lot longer than 6 nanoseconds, but this is just to give you the idea. They're both within the noise, so neither the twisting of this thing inside the protein or any other change or the energy which is released, because this releases a lot of energy that jumps between one surface, makes any difference to the protein itself. Fluctuations remain the same. And so the isomerization is really not sufficient, and we could have got all the effects that we wanted to from this by just simply twisting the dihedral and forgetting about the quantum mechanics. which is good from my perspective because I really struggle with the quantum mechanics. But that's not totally true, because what actually happens with this system is that when we actually shorten the molecule, you wouldn't dare stop it too early. When we actually shorten the molecule, what you see is that there is a change, and this is actually the potential energy surface of transferring one of the protons from the, actually in this case it is, and I should remember it's the glutamic acid and not the tyrosine, which is what was originally expected, from the glutamic acid across to the phenolic group. And in fact, that after you isomerize it and cause this slight shift in it, is that you actually make having the proton on the phenolic group and destroying this actually negative charge on the phenolic group, you actually make this a more stable state. And so if we actually look at it before isomerization, we actually calculate an estimate of the energy of proton transfer, you can see that this never becomes a favourable event either from the tyrosine or from the glutamic acid. And simply proton transfer isn't possible. After the isomerization, what we see is that still for the tyrosine, this is almost never a probable event, but you can see that for the glutamic acid, there become structures due to the fluctuations and dynamics of the proteins which actually allow this transition to be possible. Once this transition occurs, it will never go back, and this is a committed step in the process. And of course, once we actually allow it to undergo this proton transfer, you see there is a very rapid change in the fluctuation. The protein begins to unfold. And that you see particular increased flexibility in the N-terminal region, which actually matches what has been seen in NMR in terms of the changes in the dynamics after you actually hit this with a little bit of light. So what can we say about the isomerization mechanism? Okay, the first thing which happens is that on the excited state surface we see a rotation around this particular double bond. The rotation itself does not cause a transition back to the ground state. It seems to be quite happy to actually sit on the excited state, even with a 90-degree rotation bonds. But if we're actually seeing other stretches, when they get to this point, this is actually what causes us to see the collapse down to the ground state. This strain which we introduce then disrupts this interaction, which causes a rotation around of the scion. Ester. In terms of the signal transduction, which a lot of people have thought was really intrinsically linked to the absorption of light, what we see is in fact a transfer of the proton from the glutamic acid back to the charged phenolic group. This actually destabilizes this interaction, which leads to very rapid partial unfolding, and then from then on to actually the signal transduction inside itself. But that part is certainly another one which revolves a whole lot of other secondary messages which actually bind to the partially unfolded state. And then it just has a simple time loop back to the original. So I should really point out again that this work is primarily that of Gerrit Groenhof, especially with Berenson, with Groningen. The quantum mechanical parts are really due to a collaboration with Mike Robb, who is in Birkbeck now, I think. I can't remember where he kept on moving to. And it was also done. The what? Imperial.
Imperial. You got it. Unless he's moved again.
Actually, I think this is on. I think whether we went from Imperials to King. I can't remember which way, whether I had it. I never can remember anyway. And it was also a lot of the original interest in PYP was that of Helingworth at the University of Amsterdam.
Thank you.
And if you ask me about basis sets or something, you'll be very sorely disappointed.
Right.
Just something that came out of Seth's talk as well. The photoisomerization, Seth mentioned about the charge distribution getting sort of a positive and negative charge forming. I mean, you're sort of missing from some of the other two latter images the arginine that was sitting sort of on top of the ring. Do you know if there was any?
You get an increase in the charge in the ring system in the excited state. And so that's why we expect that is actually causing, if I remember correctly,
what about the two carbons that are actually involved in the the motion, I mean, is there, for example, a negative charge, partial negative charge forming on that carbon that's closest to the arginine, and sort of basically holding it up there? You said it's quite happy to sit on the excited state. I was wondering if that was sort of stabilising.
This is really not something I can answer myself, because I'd really have to look it up. So as far as I was aware, that during the excited state, that we were getting charge on the main ring system, which was interacting with the arginine. I'm not aware of myself what changes. I mean, they were calculated, of course, but I can't tell you exactly whereabouts the charge went in the system.
I mean, related to that, is this supposed to be a glenoidal structure, or is it supposed to be a phenolic structure? So it sort of comes back to the question we were talking, with Seth as well, where this sort of isomerization potentially occurs through the glenoidal structure, but here you've got sort of carbonyl along to something which essentially, is it phenol, is it quinoid?
Okay, unfortunately, I am really dreadfully the wrong person to ask this, because was it the, I mean, what we were focusing on really was just what the protein was doing, whether this was causing strain, not the details of exactly how the isomerization was occurring, other ones like that. Not me personally. Gerrit could certainly answer it, but I can't answer it. I was interested in the classical aspect.
Yeah, but it's kind of interesting because it's a similar mechanism, it appears to be a similar mechanism to what's occurring in the fluorescent proteins as well. Maybe there's some sort of uniformity across that whole family of materials.
Recently, there is a FTIR experiment that is showing that there is not at the early PG state, the glutamate is protonated, and also the chromophore is protonated, which means there is no protein transfer in the formation.
So you're saying that in the state that they're both protonated?
Right. This is something suggestion by the FTIR.
But then how would you get a protonated glutamic acid right next to a protonated phenolic group?
I don't know, but the FTIR experiment shows that this is protonated at PG state. I mean, the chromophore takes a proton from outside.
So that certainly isn't work that I've seen. I think there would be a lot of people who would want to question it, because my understanding was that the evidence to say that this was actually a charged phenolic group was actually very strong. So, but I'm not aware of the work that you've been.
Late PG state, there actually the proton is the later state, the glutamate is protonated and the chromophore is unprotonated.
Yeah, but the PG state should be the resting state. So this should be before.
Actually, there are two states. Late PG and early PG, there are two states which have blue-shifted absorption matrices. This is early. It is called early PG and late PG.
So you were saying that the first step would actually be the proton transfer after photoabsorption.
The first process is proton transfer from bulk to the glutamate.
Okay, well that I haven't.
I'm not talking. What I wanted to ask you is there any suggestion for that experiment? I mean, if you have very large barrier for this proton transfer process.
Well, I mean, in the calculations we did, that there is no barrier for the proton transfer process.
Okay.
I mean, we know that that is a relatively slow process on a biological time scale, and that just even within nanoseconds, we're seeing fluctuations which actually make that proton transfer basically a straight downhill process. So, if we give it enough time for the fluctuations, then I mean, if we see it on the time scale of these simulations, there really can't be a barrier.
Okay.
I mean, it's.
But experimentally, the formation of PG takes microseconds. So in that sense,
the formation of the partial unfolded, I understood, took
I mean, the spectral shift is due to the blue shift,
Yep.
right? So, which means that if proton transfer takes place, spectral should, shift to the blue. So, if you're saying that the proton transfer takes place nanosecond.
Well, what we're saying is that if in a very primitive way that we calculated the proton shift, there were sufficient fluctuations in the simulation to make this energetically favourable. Now, you could argue about whether the way that it was calculated was accurate enough, whether this is going to occur instantaneously once it's actually become, because it's not constantly one. It really requires a fluctuation. So there are many reasons of why our timescales may not be be correct. And I wouldn't bet one way or the other on it at the moment, not based on this calculation.
Thank you.
Hi, Anna. This is fascinating. I just curious. I don't know. So you have glutamate, a protonate. Is it could be the harnessing with protonate?
The what? Again, the only thing I can say is that when we calculated the probability from the configurations on the simulation to simply look at whether there was an energy or not barrier, is that to the tyrosine we always found that this would have been a positive, there would have been an energetic cost, whereas for some of the configurations we found that it was favourable for the glutamic acid. So for that reason we propose it is more likely to come from the glutamic acid than this to come from the tyrosine.
Okay. Another thing is this always need to finish your photo cycle.
Yes.
So I would say when protein unfold, the glutamate will release the proton, and then this chromophore should grab a proton and then form the initial states again.
Well,
so
this will spontaneously re-isomerize when the protein is partially unfolded. But this is a process which takes microseconds to milliseconds. And then, of course, that they all risk. I mean, it's actually. I mean, if in an unfolded state, you're not going to get a protonated glutamic acid, and I don't think you're going to get a deprotonated phenolphthalein. I mean, so it's only once that they refold and reassemble back into one that you end up with these interactions, which will actually allow that particular state to exist. I don't think there's any suggestion that this is occurring in solution.
So what causes it to sink then?
The what?
You said
it swims away. It swims away.
It's actually, it continues swimming. It just swims.
Well, I mean, but this is, it's a very common mechanism in bacteria. When the flagella are all going in one direction, they all constructively so will actually swim in one. Now, when the flagella reverse their direction, it tumbles because they go like a little ball. Okay, so this thing is coming always up to the surface. It floats very slowly because it's almost neutral buoyancy. But then, of course, what it does is it swims in a particular direction. If it gets hit by a photon of light, it says this is the wrong direction. I stop. I tumble. I begin swimming in another direction. If it doesn't get a photon of light, it just keeps on swimming. And that keeps it swimming away from the light. It's the same way the bacteria use to swim towards food. They use exactly the same. But what you need here, what's critical, is the timing mechanism. And the fact that this is a saturatable system. If it gets lots of light, of course you've saturated all. It remains in an unfolded, non-singling state. So it's a very simple little mechanism. Cute.
Sorry.
Yep.
You mentioned this proton nature is downhill, right?
You really want to ask me about the more lengthy.
Why don't we leave the discussion there and talk about that over afternoon tea? So all that remains is to thank Alan and the afternoon speakers. What's
the reception?
In a section entitled Biomimetics is Professor Kenji Gano from the University of Melbourne.